This is a production of Cornell University.
So as the Moira pointed out and the title says, I'm going to walk you through how it is to start a breeding program from scratch with a crop that many other breeding programs are running for 100 years or so, or even longer. So in my talk today, I'll kind of cover shortly my background of the breeding in South Carolina in Clemson University. Then I'll tell you about the challenges that I have and accomplishments in this 13-year period. And the accomplishments would kind of be designated as discovery and application, as what you will learn throughout this talk is I'm all about application. I don't start discovery unless it's going to be applied. Otherwise, I don't see the point of the discovery. And so that's how I structured the talk and basically how I live my life professionally and mostly personally too. And so some of the future things will trickle through the slides. I'm not going to have a special slide saying future, but you'll get the gist what the future is in certain points that I cover. Okay, so that's not what I wanted. This is what I wanted. Okay, so before I start, I always like to start with the slide showing a great fellow breeder, Dr. Oaky, that retired right around the time I got hired. And my first visit was to him and to Dr. Ralph Scorza and USDA and Carnesville, West Virginia, to kind of pick their minds on how do you start breeding program for peach from zero when there are programs in the United States, public and private, running for 60 to 100 years. Where do I fit? What do I do? And Dr. Oaky actually published an excellent paper covering the East Coast prunus or stone fruit breeding breeders and breeding programs. And in that paper, he listed the breeder qualities, and he said it's just an overall for breeding. Doesn't matter if it's a plant or animal breeding, and said that the breeders have to have a vision, the ability to see what's needed and what is feasible, that they have to have a passion and devotion to job daily and over the years, and the willingness and ability to work for a distant payoff. And you'll see all of these qualities or maybe lack of them through my presentation or something that I need to still work on. Discipline, you need to keep in mind what is the goal and execute the many steps that sometimes seem meaningless to get where you want in efficient way. And the final thing is the risky thing where you actually put your stuff out there for others to criticize. So you'll see some popping up these words through the slides to kind of get you an idea where these disciplines are playing the part. Okay, so why peach? Why in South Carolina? South Carolina, for most of the people, they don't know it because we are fighting with Georgia about the peach rights. But here you can see where we are in the United States. We are second in nation for peach production, right behind California for fresh market peaches. And even though Georgia has the rights to call themselves a peach state, they are more like blueberry state lately than the peach state for the amount of the production. So we have about 18,000 acres or so under the peaches, and it's a #1 industry in our state. So no wonder that growers wanted the breeding program that would address the needs of our environment. So breeding program was rejuvenated in 2008 when I got hired after 25 years of dormancy. That basically means there was no crossing going on when I got hired into the position. So what is it that I found out when I got hired in Clemson? So one of the great things about Clemson, and luckily we still have that, is we have a great team of people that are all addressing the same crop from different points of view. We have horticulturists, pomologists, pathologists, entomologists, virologists, geneticists, but at the time they did not have active breeder that actually would pull all this information and try to deliver genetic solution to growers' needs. And so that's where I fit into the picture. And we still kept all of that team together, which we replaced certain positions, which is really nice that we did not get rid of them after people retired. And so we now still are trying to make the best possible solution for our growers. So what I found in Clemson, with all that great group of people, they had an excellent germplasm to work with and facilities. They just did not have the breeding material going on, like crosses and in different level of selection. So when you inherit a breeding program, you basically inherit a program where you have all kinds of stages that you'll see through my talk of the level of selection and progression of the material. I did not have that. So my first cross I made in 2008. With peach, we wait for 3 years to see the fruit. So but sometimes 30% of them can produce the fruit in the second year. So the first fruit I could evaluate was in 2010. So that was like first 2 years, which I cherish now. And at the time I was like, okay, when it's going to be like, when I'm going to see my fruit that I could use to learn about the germplasm, about the problems. I come from Eastern Europe, where I was assistant professor of fruit breeding and genetics, and I did apple and peach breeding, but the problems are not the same. The climate is not the same. So that was a big eye-opening. Something that we didn't even worry over there is a big problem here in South Carolina. So that's what I brought with me. I had a conventional breeding experience, and I was trained to do molecular side, but never had the classes. None of my education was molecular, which tells you something. If you have a solid genetic knowledge, everything else is like a cookbook, or you have great people that you can collaborate with, which you're going to see through my talk, that can solve those issues for you. But if you don't know what you need in your material, none of the great tools that exist right now that are getting cheaper and cheaper will get you where you want to be with your program or development. So those are the big things that I always try to kind of instill in my students to understand. So in 2008, when I started, we had a great community resource called Genome Database for Rosaceae. Peach belongs to the rosaceous crops, and we had an excellent one-stop shop where you can go and get an idea of what's available, markers, linkage maps, QTLs detected, and knowledge. At that time, that's all we had. Most of the markers were rapids, RFLPs, which you probably don't know even what it is, or it's like some ancient history. SSRs, which still get used a bit. Well, some of the linkage maps were really low density and needed improvements. So this is what we have in 2021. In between 2008 and 21, we got peach genome sequenced and now have many more other peaches sequenced genomes. We developed some tools such as snip array, and we improved those tools. Many more markers, linkage maps, transcriptomic and metabolomic data available at QTLs. So none of that was available when I started. And that's where I kind of wanted to be in 2008, you know, but you just have to put the work down. Lucky for me, most of my work coincided with the Rosebreed project being funded for 2 rounds. And most of the things we proposed in that project was what I was hired to do. So when I got hired, the first thing was to go and talk to growers. Okay, so what is it that you want? What is the ideal peach that I need to create for you that you don't have right now available for you? Because if you look into the patenting release, there are about 500 peaches available for growers to pick to grow. The problem is when you grow them in South Carolina, you encounter some problems. So what they wanted me to do is the standard: yellow, flesh, firm, melting, freestone with blush 50% or higher. They wanted the soluble solids to be at least 9 bricks or higher in early material, which always doesn't taste really well. Then reduce the split pitting in the early material and extend the ripening season if I can, and fix the size of the fruit in early ripening material. And of course, introduce the resistance to the diseases. That was the niche where my program fit, where other programs, not that they didn't want to work on it, they did not have environment to see those problems on their material. So when we would bring the peaches from California to South Carolina, they would fail miserably because they were susceptible. The humidity brought so many disease and insect pressure issues that they've never seen in California with the dry climate that they have. And so that's where I fit. And so in the meantime, I basically figure out that I need to also work on restricting the chilling requirement because of the changes in the climate. When I was hired or was interviewing for the position in 2007, there were no peaches in Southeast. There was late spring frost wiped out the whole production. So then also I wanted to extend the life of fruit on the tree to reduce the number of harvests for growers and to increase the quality of the fruit. Because the fruit, unfortunately, that you buy in the store is not even close to what you get when you pick it from the tree. So we want to kind of close that gap. And also nutritional quality and flavor were some of the things on my mind, and many others that you'll see trickling that I'm not going to even mention. So based on that, the initial goals of the program were, of course, develop a high quality, productive, disease-resistant peaches for Southeast environment. And of course, the main focus was combining all that great quality with the disease resistance, and two diseases that we kind of focused on, the bacterial spot and brown rot, that are actually creating a big problems for our growers. In the meantime, as I already mentioned, I kind of expanded that portfolio and tried to pull all other traits in, because you'll understand later when I go through the how many years it takes to develop anything and create any variability in your material, that you need to kind of be a magician and visionary and see 20 years from now what growers would need. Because when they come and ask you, it's now, they want it tomorrow, and you know that's not working that way. And as Moira mentioned, I kind of got dragged into the rootstock breeding as well. And I'm on that side just as a breeder. I make crosses, I do genetics, but I leave the rootstock evaluation to the experts, because that's now becoming a big problem. If you know that trees compound from rootstock, so roots are from different plants than the scion, the above ground part. So if the rootstock is not fitting and cannot survive on a certain soil, no matter how good your scion is, there's no production. And so that's how I kind of ended up leading the project. You might say it's a funny story, but you go where the need is, again, what the growers need. So here is just to go through some challenges. I already mentioned the climate, consumer demands. We as a consumer, we flip 1 year to another what we want or how we want it, lower pesticide input, better nutritional quality. And so you kind of have to predict that. Also the producer needs, they want sustainable production, cost-effective disease resistance. And one that I found out is the hardest one is the retail education, because we in the meantime got so many different types of peaches that nobody pays attention to, nobody educates how you should sell them, how you should tell the consumers, okay, this is different from what you used to. In apple, you name the apple cultivars. That doesn't happen in peach. You don't go into the store to buy Granny Smith or Cosmic Crisp or Snapdragon or whatever name you know you like. You get a big basket with a mixture of peaches. Peaches last only 2 weeks. So you have mixture of several cultivars. Our growers grow in around of 40 to 60 different cultivars throughout the season to cover the whole season. So there's work to be done there. This is kind of the bird view of what my program is doing or trying to do. And some of the things I'll touch today, and many of those will kind of be left over. And so I just wanted you just to have an idea how many things played a part in just creating one tree that has all these characteristics that we want to combine them. This is where the magic happens. This is our farm, and you can see that the location is ideal for the fruit growing. It's surrounded by the lake. And so with arrows, you're gonna see later on where I have my fields. We basically have 3 sections that are already cleared. Section here, here that goes into this, and here is the main building with the labs and the section that's here. All of this land here and here also belongs to the farm. We just did not clear the woody area for it to be used. And you can see it's about 200 acres of land, and 90% of that is under the peach, and the rest is under the different fruits for mainly student education and pleasure for us to eat different fruits, whatever we can grow. And so I'm one of the biggest user on the land because I plant my hybrids, I keep them there for 2 fruiting years, and then after that, I graft them, move them to another location within the farm where they are trained and kept similar to what the growers do in the regular production. And then I look at them for 2, 3 more fruiting years there. Then we do another selection, and then we move them into so-called replicated trials. So these arrows kind of showing you where I had at the time, because this drone picture is no longer accurate. You know, things are moving on a yearly basis, where my things were at the farm. And so like here is the isolated one where we try to grow without fungicides to see if the brown rot tolerance that we developed into the material is actually holding up, and how much we can actually reduce the spray. We'll never be able to eliminate the spraying because it's just the type of the fungus that will always be there. But if we can reduce the number of sprays to maybe just before harvest and not the whole season, that's going to be a big winner on the side for growers and the consumers. So I kind of alluded to the challenge of the time. So this is the timeline of the breeding program. In the year 1, we pollinate, then we stratify the seed because they need a cold treatment before they're going to germinate. We plant them in the greenhouse when they overwinter. If the material is the mother tree, is the early ripening, at the time of harvesting the fruit, embryo is immature. So you have to put them in tissue culture to give them the nutrients, keep them under the 4 degrees Celsius to give them the cold treatment, and then raise the seedlings, and then plant them in the greenhouse. They overwinter in greenhouse, and the next year, springtime, we plant them in the field. They grow there for 2 years, and then we start evaluating the fruit. And so here is some of the numbers that I've been accumulating throughout these years. And this is where I am right now. I'm in the replicated trial stage, where you can see, starting from all of these numbers here, where it is about 1.7% of the evaluated trees end up to the 2nd level of selection. And then out of these, we select those that go into the replicated trials. Now, right now, I have 10 advanced selections in replicated trials that are at 7 locations throughout the Southeast. And they this summer, we had a 2nd year of data collected from those. And some of them are with the research stations with the universities. Some of them are with growers. And out of all of these, one stood out for the growers that they really like, and that's going to be my first release cultivar. So yay, I came to the finish line in year 13. I mean, it's not released yet. We are collecting the data because it takes time for everything you learn through the talk, but that's so fulfilling to know that all that planning and work actually resulted in some excellent cultivar that's bacterial spot resistant, which is the big deal for us. Okay, so how do we do phenotyping or selection in our program? We do it in a field. I basically walk the field and eat the peaches all day long, and then jog some data in it. You'll see, I'll mention some of the gadgets that I've been trying to test through my life as a breeder here to go to something that's much more manageable. And then we bring the fruit to the lab and we run the lab tests to determine different qualities and then try to merge all of that data into one to help us decide which ones are worth moving to the next level of selection, where we invest the money into grafting them on the rootstock, training them as trees in the regular production, and which of course costs the money, it costs the labor. What I was hired to do is to merge the conventional breeding with the molecular tools to speed up the selection and be more efficient in selecting. So those are the two different things. You want to eliminate material that has no chance of having the combination of traits you want early on, so you don't waste the time and resources in evaluating. And you can go and follow the slide. I'm not going to read. The final idea was to increase the efficiency. And that's something which this cartoon here in the top right shows that we try to do with the first rose breed project. Just look at the fruit quality, define what is the key quality, fruit quality trait that growers are after, try to develop the markers to try to at least predict which parent combinations we should use to get that quality. Because more so than often than not, when you combine two great looking parents, the children are not anything close to the parents. It's always the second generation or the third, even in a human population. I always say to my mom, why am I like this? I mean, you looked so great when you were a young girl, and then my daughter is like my mom, you know, so it's keep generation. So it happens with the plants, guys, too. Nothing is different there, too. Sometimes the great parents are just disappointing. And so you have to kind of know more under the surface what's going on. So what we try to do, when I started, there were great level of maps and known locations of the traits and genes, but not many tests, not even one. And to get from that, knowing where your gene is responsible for certain trait, to the tool or test that you can apply and trust. Believe me, we breeders are rare breed. You don't trust lightly because it takes a hard work to get that seedling. And so you need to really trust that test or information you're getting to kind of chop that tree and remove it, because once it's gone, that's it. And so here is some of the tools that we were using in phenotyping and the countless number of trays, and when we did to develop 5 or 6 tests for fruit quality within rose breed 1. Then under the rose breed 2, we said, okay, now we have some tools for the fruit quality, but we really need the disease resistance. So I kind of doubled down in the first rose breed using the tools that we developed for uniform genotyping, phenotyping to already start working on the bacterial spot. And here is the picture of my PhD student at the time. He basically found out that there are 2 genes and on 2 regions in the peach genome that are responsible for tolerance or susceptibility to bacterial spot in fruit. Because you can see here on these pictures, what does bacterial spot do to the fruit? It's not bad fruit to eat, but nobody's gonna buy it or nobody's gonna reach for that fruit to eat. It also can defoliate the tree like here, and that happens in July. So trees are basically wasting all of the energy that they need for the next year flowering to push another round of leaves, because they basically get defoliated in the middle of the season. So the growers told me, we don't care how the tree looks as long as it doesn't affect the next year production. We want a clean fruit. So that was my kind of first focus on. And so we developed the SSR-based test. Wasn't perfect, but at least it allowed me to select the right parents and select some level of selection for the hybrids. In 2010, after 2 years of crossing, when I got mediocre like 1,000, 1,500 seedlings, I said, okay, that's it. Anybody who's willing to go out in March, let's go emasculate. And I ended up with 15,000 seedlings. Well, never again. Because 2 years after what I did, I was just walking down the rows and eating peaches. And by the time I got to the end, there's a time to start all over again because they don't ripe at the same time. And so I said, okay, no, I'm not going to do that until I have the marker to eliminate something that's susceptible for the bacterial spot, I won't be able to cut the numbers because everything else, fruit quality wise, it can go here and there, but for me, make it or break it is bacterial spot tolerance. If it doesn't have it, I just don't care what any other traits it has at the greenhouse stage. So in the meantime, we perfected the test, and now we have a SNP-based test with a CUSP application. We run it on an endpoint PCR machine. Those that do this will understand what I'm talking about. We don't need the real-time point machines. We just need it to read the plate. We use the crude DNA extraction, so the price is really low, like 10 cents per plant. And that's the test developed by Strawberry Group. So you are hinting to the family and borrowing from other and culinarity symphony and what's going on in the rosace later on. But that's the beauty of it. You look into within the family that your plant species belongs to, and you can pick many things and tools and ideas from others, and you don't have to reinvent the wheel. So we were really lucky that that test actually proved to be excellent. The University of Florida group developed the test for there. It works with a high resolution melting, with SSR, with the SNP genotyping, and it's a crude extract. That's just the beauty of it. And it takes just the two homemade buffers that you have the chemicals lying around in your lab to make it in one day, and it's just 2 steps, pipetting, overnighting in the +4. So it's really, really easy and worth trying for all of the students and colleagues that are listening. I urge you, if you have an issue with pricey extraction, and if it's just for the quick marker test, try that test. It's really, and you, I mean, while I was bragging about that test, you could have read all of the numbers in the meantime. We are right now testing over 6,000, 5,230 seedlings were stratified. And one more thing that I wanted to point out, what we do even now to shorten the period of the youth, we apply the hormones to break the dormancy. So basically the 5,000 seedlings that we already tested, growing now and will be planted at the farm, our climate allows me to plant them in the same year we actually made a cross. So they overwinter at our farm, and next year, so I shaved the 1 year from those 3 years before I see fruit in actually making the decision on which fruit hybrids are gonna be advanced to the next level. So I was very pleased to find out when we developed the SSR test and later on the SNP test, to go back through my program for anything that I selected based on the phenotype, just the quality of the fruit, that I already have some great series of material that covers the ripening season. That's whatever I listed there as an ideal peach. And so, which meant, my great colleague, John Clark said, it's serendipity. You never know what you're gonna get. So you have to kind of have that in mind, not just trust the tools, or so I'm kind of in between. I'm developing the tools. I trust them a bit, but not completely. I still kind of sneak something. As long as I have a space, I'll probably keep more than what I need to. But that's just like my children. I cannot just get them out, because once they're out, that's it. So that's the easy story. My students would not say so easy for the bacterial spot. Well, now we get into the fungal disease, brown rot. And that one is the nasty one, because it starts with the flowering and it goes into the fruiting. So the whole season growers have to spray, and then even goes into the post-harvest, because if you have one fruit that's diseased in the storage, it's gonna ruin whole basket or whatever crate they have with the peaches. So we did classical, phenotyping, and then we developed some families, and then you can see here, like the average for the family is nothing exciting about, but within the family, we had some that were completely clean. And so the solid bar color means wounded, means the flesh tolerance, and the striped ones mean the skin tolerance. So we would wound the fruit to allow the entry point for the fungus and to see how flesh tolerates infection, and also try to have the fruit with the perfect skin without the micro cracks, to make it hard for fungus to penetrate to see how well they're gonna tolerate. And some of these actually end up being with the right type of flesh and with the right level of the blush. The problem that we had is that the source of the tolerance comes from the landrace from Brazil. Well, that thing looks more like this, but no redness. It's clingstone, which means that the flesh is clinging to the pit, which is what people don't want. They want to like split and toss the pit and eat the flesh. And then it's also non-melting. So it has kind of a rubbery feel when you bite into it. And those are perfectly fine peaches. They're just different from what people are used to. And especially for this region in North America, they're more accustomed to the melting type of flesh that basically falls apart in your mouth and fully juicy. And so that's kind of an educational side that we need to do. Well, when we did the genetics on this, it's not a clean story about, like for the fruit for bacterial spot. We had signals all over the genome and we could not get a clear picture. So there was no way for us to develop some SSR or SNP-based tests to run. And then we use genomic prediction to see if we can pick up the best material to move forward and overlay that with the bacterial spot fruit to kind of go in parallel for both diseases. So since last year, my crosses are designed based on the both information. So I put the point in making the parents the best possible chance to create that combination that I need. And you probably heard, if anybody before talked about breeding, it's a number game. So the more progeny you can get, the better the chance you have to get that combination of traits that you're going after. So I'll just cover this really briefly. We also look into nutritional composition. If you heard of people talking about peach is genetically really uniform. And you don't see much variation on the genome level. Well, on phenotypic level, when you look at them and taste them, there's a bunch of variation, and we still have a work to do to figure out what is it that gives that variability. So we found a great variation in nutritional compounds accumulation, and we did some mapping. We tried to do the genome-wide association because of the wide panel and some biparental mapping. We also looked in the specific ones into carotenoids, and also across the whole prunus family of stone fruits, in this case, apricot, plum, and peach. We wanted to see what is it. Actually, we know that the flavor and aroma you get in a white flesh peach is due to carotenoids that are not spent in giving yellow flesh color to the yellow flesh peaches. And it's all due to one gene. And so if that gene functions, you end up with the yellow. If it doesn't, you end up with the white because the carotenoids don't get processed. And so we wanted to find out, okay, what is the variability of carotenoids in the yellow flesh material, then compare it to the plum and apricot to see if it's the same or different. And then did some aroma profiles of extremes to see, can we pick some aromas to actually bring that great aroma white flesh peach has into the yellow flesh. And so it's still work going on, but there were some really funny and interesting things that we found because we analyzed 300 peaches in 2 seasons in really expensive and laborious profiling on carotenoids, and we got completely different results than what was published for years because everybody was doing 2 or 3 cultivars because it's so expensive and so labor intensive. So we are really looking forward to see what's gonna trickle out down the pipeline from this work. So looking into all of this, you can see that many traits get mapped and many of them actually land on the same chromosomes. and in the same regions. So we call those so-called hotspots. And so what our idea is to define these blocks that are not recombining, that always are inherited together, and then understand variation of each for this block, how many different variations it has, and what does it mean phenotypically. To basically do, this is for chromosome 4 cartoon that we figured out has 14 blocks that do not recombine, and then do the design approach to cherry pick for each of these different blocks, which one we want in the ideal peach. Of course, easier said than done, but since I started my position, almost every single crop in the Prunus genus, economically important crop, has a genome sequenced. The only one that doesn't is the tart cherry, because they're, polyploids, so we kind of put them aside because they're hard to work with. And so I'm happy and lucky that peach is diploid, self-pollinating, have all kinds of nice features to work with. So those 3 years of waiting for the fruit doesn't seem that bad when you look in some other fruit crops that are longer. So with these genome sequence, now we have ability of actually comparing the regions in different genomes and seeing if the same traits are landing in the same chromosomes. And then if they do, maybe that means that the traits that are mapped in one crop but not in the other will actually point to you in the region in the genome for this other crop. So you don't have to do all the biparental mapping, all of this legwork to get to the point where you can just compare them and get an idea, okay, do I have variability? Do I have more genes in this region? And so this is what we've tried to do between sweet cherry and peach, because within the region on chromosome 4, from 7 to 20 million base pairs, we have mapped the same fruit quality traits. And so peach has many more traits mapped in this region than cherry. So we could kind of go back and extrapolate. Cherry has one big one that we don't, which is the fruit size. So, we can kind of use the family and resemblance between genomes, which we call collinearity and synteny, which is even on the level of rosaceous species very high, to kind of inform ourselves in what is the possibility. Because as a breeder, you need to know, are you in a bottleneck? Is there nothing you can do to break that solid block that gets inherited and carries the bad traits that you don't want? You need to know that, so then you don't waste your time and you try other approaches to kind of do the recombination. So one of the things that we used, if you work with a model species, there's all kinds of mutations and stuff that people accumulate. Arabidopsis is one of the great examples that you can find all kinds of answers. We don't have that much in the fruit trees, but there is one great mutation family in Japanese plum. So all of these listed here are the 5 spores of the same cultivars, and they were picked up because they ripened at a different time. So in the fruit trees, with apple being one of the lead in, is you can just walk through the orchard and you can see the branch having more red fruit, bigger fruit, earlier ripening fruit. You just cut that branch, you vegetatively propagate it, and there go, there's a new cultivar. So many apple cultivars were actually named like that. So we have the same similar situation in Japanese plum. And what was not known at the time is that not only they ripen at the different times, they also have a different way of ripening. So peach fruit is climacteric, which means you pick it from the tree and it continues to soften and ripen. But there is a non-climacteric or suppressed climacteric type that we found here. And basically, when you apply external ethylene, which is what they do with apple to basically keep them in storage and then apply that external ethylene and then ripen it for selling it in the store, with these plum, we encounter that there are some that do not soften ever to the rate of softening of the regular or the original cultivar. So we were looking at the genomes and transcriptomes of those and trying to understand if we can kind of apply that to peach, because peach flesh can be softening, fast softening, non-softening, stony hard. Stony hard meaning even if you add external ethylene, it's never going to soften. And so there are some issues with maybe making them transportable, more tolerant to processing in the packing lines and all of these things that we are trying to look into that. You can imagine how many data I collect. All the genotyping, phenotyping, profiling, all of this with different students, different colleagues that I collaborate. So breeders are great, how do I say, in the like people that assembly group people or like people around themselves or expertise around themselves. I cannot know everything, so I'm a great person to know who knows and who I can actually collaborate with to help me. One of these guys is Dr. Sasky, which is a great translational genomics, and poor guy works office next to mine. So it's like, I'm always there asking for something because I'm like hands-on. I like to do it on my own because if you work with the people that are bioinformatics only, we sometimes do not talk the same language more often than not. And so we are getting better, but if you understand how they think, it helps a great deal of actually making faster improvements to whatever you want to accomplish when you want to use some tools. And so I already mentioned the Genome Database for Rosace, and they basically helping me by developing databases where I can store all of that data sets. Then something that was developed for a wheat breeding program, I actually utilize in mine. It's called Fieldbook app, that I go into the field, collect the data, and it gets into the breeding information management system, what BIMS stands for. And then it's immediately at my fingertips to make a decision, even on a daily basis, as soon as I collected something in the field. So in 2010, when I first looked into my trees and I was waiting for them to first time have the fruit, it was a wishful thinking. Now it became the reality. Now I know many things about those trees before I ever see the fruit, which actually cuts down enormously the waste of effort that I was putting into trees that I would just knock out and never do anything, no advance of them. Of course, I was so pleased to see that among the 2014 crosses, I actually have a brown rot tolerant material that is freestone, melting, yellow flesh, almost 100% redness of the skin. And I also have a bacterial spot resistant material, similar fruit quality, and one with both of them in the same. It still needs improvement on the fruit size, but you know, the hard part is there. I already have 2 tolerances in that material. And growers are interested in trying to grow them for the organic type of a production. So something that I actually did not think about, you know, the vision part, you know, I was like, okay, no, that's not for me. You'll never be able to grow organically fruit, but then just might. Of course, breeding program at university, externally funded. So that's all the grants and stuff, major ones that were supporting the work. And of course, the South Carolina Peach Council and Department of Agriculture, and many, many, many people. Not just faculty that supported me and helped me, but students, starting from undergraduate and graduates that were working on all of these nice things that I'm standing here and showing off for, that has an army of people behind me that helps with actually achieving this. So that's what I had. I hope I didn't go much over the time, and I'm open for any questions you might have.
Do we have any questions for Ksenija?
Yeah, it's great talk, Dr. And it seems to me focused on just the science, but we know the quality, disease resistance, a lot of confirmed by stocks. So do you have any plan to move towards the sort of breeding in terms of stock? Pay attention to that.
That's a great question. One that I would quickly answer. I don't care. If you're the rootstock breeder, you need to make a rootstock that does not ruin my science. So that's the funny answer to the question. But, and it's a reality. It takes 15 to 20 years to develop the science, up to 40 to develop the rootstock. So Not really good for the rootstock breeders, because not only that they have to have all the traits in the rootstock, then they have to test on the major cultivars to see what effects that rootstock does and that make sure that it's not detrimental. Because if you think of it, once you produce the orchard, if you have the winner rootstock, you're done, because everything's going to be planted on it. But then if 5 years into the planting the orchard, there is a major flaw that like causes all of the trees to die or something, you basically run somebody out of the business. So that's why rootstock breeding is much harder, because they have that additional level of testings. So now that I was dragged into the rootstock breeding, you know, now I'm paying attention, you know, because now we want to develop the rootstock that is tolerant to the replant diseases. There's a bunch of things that affect them when you replant them. And if you think of it, in South Carolina, we do not have a new land. We are planting over and over in the same land. And so our Milaria project that Moira mentioned that got just funded, it's a crucial problem for peaches in Southeast, for cherries in Michigan. It's slowly creeping up in California as well for grapes, for walnut, anything that's long-lived. Armillaria is a really interesting fungus that it doesn't, there's like more than 30 different species of Armillaria. They're adjusted to every plant because it lives in the soil, and it's major under the forests in like Colorado, Oregon area, and they learn to live together. We need to find out how, so we make them live together if we cannot get rid of it. Yeah, but that's an excellent question, and that's we never actually asked, is there something in a scion, you know, towards the rootstock? We always look, okay, that rootstock, you know, delays the bloom, or, you know, makes it earlier so they get, you know, killed by the frost in the spring. But sometimes maybe there is a backtalk from the scion that we just don't look at.
I have a follow-up question for that. So do you think that given the age of genomics, and now that you have all of these, you know, having a pan-genome basically, if rootstock breeding can actually kind of follow and have shorter cycles of selection, given that you have more genetic information now?
Actually, we are hoping to achieve that, because it's like opportunistic mostly. Now that the pricing is not that high, and that we learn that we have the genomes, we compare them, that there's so much similarity. Now many so-called orphan crops are getting picked up, and much faster to the level of these first ones that started, like peach was chosen as a representative because it was diploid, you know, we had a dihaploid individual to sequence the genome. So all of the things were working forward. But now cherries, we have several genome sequence for cherries. Now in the rootstock, we are doing the same, sequencing genomes, doing the comparison, trying to find the markers, and then understand, because most of the things for rootstock are happening under the soil, you know, like and one other thing that I didn't even mention is just the root environment, you know, mycorrhiza, what's going on there? I mean, we did not even touch that. There's a whole line of colleagues that are working on it that we simply have no way of bringing together because it's so expensive, and all of the funding that we are looking for, which as a president of NIPB, I'm trying to change and to raise awareness what the breeders need, that we need to kind of open up the bigger lines of funding, something like NSF has, but for application type work. Because really, when you see how many problems we have, you need the whole picture, not just focus on one line and then ignoring what's going on the other research that can actually solve your problem. And I think there are
questions in the chat.
Questions. Okay, so let's go back. Oh, why don't I have a test site in Georgia? Great question, Mark. Because our main growing region is so close to Georgia border, and the climate is almost exactly the same. And what's left of the Georgia peaches is left on their northern border next to South Carolina. That's why there's really no reason for me to plant the trees there, because the environment and everything I need to see, Georgia growers can see in South Carolina. Yeah, great question. Okay, so yellow peaches often have a more tart taste than white peaches. Do you know which gene determines peach fruit acidity? Are yellow flesh and fruit acidity highly linked in peach? We do have the gene for acidity in peach, and no, they are not linked. We have both white and yellow flesh peaches with low acids, with medium or high acids. And the tart taste, that just depends on what, what you have in the market. Mm-hmm. So that's what people expect. So that's one of the things when you're breeding and you want to change the people's perception. When you were a kid and you bite into peach, you want that memory from when you were a kid, and then suddenly that's not it. What is it? You know, what am I eating? And so that's the thing. There are yellow peaches that are not tart, that are excellent. And so for me personally, I don't like low acid peaches. I just cannot. I don't like the peach where I have to go and drink water after I bite into it. And there are some of these that are like pure sugar. Mm-hmm. And I don't like, I like the hint of acidity, just enough to keep me from actually going after water after I bite into the peach. An excellent question. Okay, based on your experiences and lesson learned, do you have any suggestions for the young breeders who are just starting their careers? Oh, many. How much time do I have? So what I can tell you, be open and accept to be criticized. I always look at the criticism as a great lessons learned, and I never get it personally, because there are so many things that breeders know and accumulate that it's never gonna get published. This is not something that you publish. That's something that you learn and then you just apply. And so the first thing I did is just reach out. Breeders are excellent community. I know we get with the PIs, like protection, intellectual protections and stuff, and exchange of materials. We found a way around it. We do MTAs, we do certain things, but we are still a great group that collaborates. And because it's such a long period of time, we are so happy that somebody young gets the position. And so when I got the position, everybody was so excited in the peach world, because they were finally great, because at the time, North Carolina breeding program stopped breeding. USDA Byron, Dr. Oaky was about retirement. Dr. Scorza in Carnesville was few years shy of retirement. He retired. Now I don't have information that they replaced the breeding. They don't even replace, like they had several breeders through course of the years retire, and we did not replace them with the breeders. So it's really a great community. And you just, if it's the program like mine, where you kind of jumping in the pool with big sharks, so to speak, private and public, you have to find a way to earn your spot. So you find something that nobody wants to work on, because the low-hanging fruit is already being taken care of, you know, and so now the harder things are left, and you have to kind of pick your poison, you know, and try to work with the group and with the system that was given to you. Yes.
So there's one quality attribute that's super important to my wife, which is the fuzziness of peach. And she does not like super fuzzy peaches. And she'd always prefer a nectarine.
Look at me, I'm just, I don't know if people can see me, but you can say fuzzy, and I can always feel them on my face. Because
some people get a little bit of allergic reaction to it. So what do we know about, because you didn't mention fuzziness as one of your phenotypic traits, or one of the markers that you're looking for. How important is it? Is fuzziness in your breeding program? And do nectarines come into play at all? And do you ever see spontaneous mutations towards nectarines?
That's a perfect question. Because when you get hired in
You may have to repeat the question. I'm not sure if we're making it or not.
Okay, so the question was about the fuzziness. Is that something that I look into my breeding program, how important it is? And I said that it's a great question because when I got hired into position, it was peach, yellow flesh, melting, But then you have to be tenured, you have to get your position. So you do what you hire to, but you always can sidetrack and do some other fun things. So I have nectarines. The problem with the climate where I'm breeding is that nectarines don't do well because of all the humidity. Fuzziness people don't like, but that protects peach from many issues of insects and the diseases. And my brain stopped working. How the growers process peaches after the harvest, they get rid of the fuzz. So it's not as important, but that question is actually excellent because recently I had a talk with somebody and they were asking about that and said that they noticed, and I noticed in my program, that they're getting less and less fuzzy. Nobody actually worked because it requires a special equipment. You have to look into the thickness of the fuzz of each of the, it's like a hair on your face, some is more hairy, the other ones are less. Same with the peach. And I have the one that growers like, called SC1 right now. We still are looking for the name. That one has almost no fuzz. So I have a fuzz scale from 0, which is nectarine, to 9, and about 7, you just don't want to touch that. No, not even bring it next to your face. And there are many more that are 1. I mean, you would think that it's a nectarine, and it's not, because nectarine is shiny and it's such a fine fuzz that you don't mind biting into that one. But because of the way the peaches are processed and displayed in the stores, you could not recognize what's the nectarine, what's the peach once they're done with it in the packing line is what I was looking for. Yeah. Okay, let me see what the next. As you implement marker-assisted selection for more and more traits, it drives up the need for larger seedling, populations. So now, as you begin to implement genomic selection for more quantitative traits, will you need to further increase population size? And do you have the capacity to multiple traits with genomic selection? Finally, okay. Looking forward to seeing me there. Okay, so that's an excellent question. So what I've seen now that for 3 years, I'm using the markers for bacterial spot, very soon I'm not going to be using it because I'm using the parents that are homozygous for the 2 locations. So everything is homozygous. For certain cross, I don't do the markers. So now I'm kind of moving to another traits. And then the size of the population, as I said, we would want to have the largest that we have. But think of this. Peach fruit is just 1 seed. So I make a cross, it's a 1 seed, 1 plant. We emasculate, pollinate about 2,000 to 3,000 flowers on the tree. We are extremely happy if we get 200 plants out of it. So it's really hard. In the process, there's so many levels where they don't germinate, die, something happens. So it's really, really hard. So what I'm trying more to focus with the genomic selection is kind of look into the parental selection, to be honest, more. And then as I get crafty and find out how I'm going to fund this, I might actually move into doing it on my seedlings later on. But right now, bacterial spot is my main one. And then we are right now testing the chill requirement marker because I want to constrain them between the 600 to 700 chill hours. And what I'm working also researching the heat requirement. I want to raise the heat requirement. So they'll sit there, get their chill requirement satisfied, and they won't bloom until they get their heat. That's our problem right now, that we kind of have to have both. Control your chill where you want it, but have the heat really high so they don't bloom until you actually want them to bloom. Okay, and that was the last question on the chat.
This has been a production of Cornell University, on the web at cornell.edu.